Hello friends, in this video we are going to discuss about differences between generalized transduction and specialized transduction. Generalized transduction occurs by virulent phase. Specialized transduction occurs by temperate phase. Generalized transduction occurs during lytic cycle of virulent phase. Specialized transduction occurs during lysogenic cycle of temperate phase. In generalized transduction, virulent phase penetrate bacterial cell and enter lytic cycle. In specialized transduction, temperate phase penetrate bacterial cell and enter lysogenic cycle. In generalized transduction, viral DNA is not integrated into the bacterial chromosome. No prophase is formed. In specialized transduction, viral DNA is integrated into the bacterial chromosome. Prophase is formed. Hence, in generalized transduction, viral DNA begins to replicate immediately in the bacterial cytoplasm, and in specialized transduction, replication begins at a later time. Therefore, in generalized transduction, bacterial cell lyses quickly. In specialized transduction, bacterial cell does not lyse quickly but is capable of surviving for several generations. In generalized transduction, bacterial DNA hydrolyzes into pieces, and in specialized transduction, bacterial DNA is not hydrolyzed into pieces. In generalized transduction, any bacterial DNA fragment is randomly packaged into new viruses. In specialized transduction, bacterial genes adjacent to previously incorporated viral DNA are packaged into virus. So, if we talk about generalized transduction, virulent phase infects bacterial cell, penetrates its genetic material and immediately starts the lytic cycle. Also, in generalized transduction, bacterial DNA is hydrolyzed and new viral particles are formed. In some of the viral particles, bacterial DNA fragment is packed, and this packing of the bacterial DNA fragment is random. That is, bacterial DNA fragment is packed in the viral particle randomly. Or we can say that any bacterial DNA fragment is packed in the viral particle. But if we talk about specialized transduction, then temperate phase infects bacterial cell, injects its genetic material, and this genetic material is then incorporated in the bacterial genome. Bacterial DNA is not hydrolyzed. Also, lytic cycle or the replication of the virus do not start immediately, but it starts after several generations when bacterial cell is induced. And once it enters the lytic cycle and new viral particles are formed, then these viral particles consist of part of bacterial DNA and part of viral DNA. And bacterial genes which are present in the viral particles are specific. Only those genes which are adjacent to the integration site of the phase DNA are excised and packed in the viral particle. So, it is not random, it is specific. Hence, in generalized transduction, some new viruses have no viral DNA. In specialized transduction, some new viruses have both viral DNA and bacterial DNA. Therefore, in generalized transduction, error is in the packing of the viral particles, while in specialized transduction, error is in the excision of prophase or phase DNA. Hence, in generalized transduction, transfer takes place due to mistaken packaging during phase assembly, and in specialized transduction, transfer takes place due to excision of prophase with nearby DNA genes. In generalized transduction, any segment of bacterial genome is transferred. In specialized transduction, transfer is limited to specific genes or loci. So, what we can say that in generalized transduction, some of the viral particles have only bacterial DNA fragment, and these bacterial DNA fragments are random, or the packing of the bacterial DNA fragment takes place randomly. So, here the error or the mistake is in the packaging of the viral particle. But if we talk about specialized transduction, viral particles have both part of bacterial DNA and part of viral DNA. So here what we can say, there is error in the excision of the prophase or the phase lambda DNA. Sometimes excision of the phase lambda DNA is incorrect, as a result of which some of the genes that is adjacent to the integration site of the prophase is excised and part of the viral DNA remains integrated with the bacterial genome. So here the error is in the excision. Also, the bacterial genes which are packed in the viral particles are specific. These are not random. For example, if we talk about phase lambda and E. coli, then during the incorrect excision of the prophase, part of the phase lambda DNA remains integrated and bacterial gene adjacent to the integration site of the prophase is excised. In this case, GAL or biogenes of the bacterial DNA is excised along with the phase lambda DNA. In generalized transduction, transfer between donor to recipient bacteria is between different strains, and in specialized transduction, transfer between donor and recipient is between closely related strains with shared site. Frequency of generalized transduction is relatively higher. Frequency of specialized transduction is low. Generalized transduction is less suitable for precise mapping studies. Specialized transduction is useful for precise genetic mapping. So, application of generalized transduction is in gene mapping, mutagenesis, to study linkage information and to compare genomes of two different bacteria. An application of specialized transduction is in isolation and insertion of genes of choice. Application of generalized and specialized transduction we have discussed in detail in previous video. If you want to understand it better, you can click the link given in the description box. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.